So we don't just need to kill bacteria, there's all sorts of microorganisms that cause infections in people. So biofilms are really tough to get rid of. Fungi, protozoans, helminths, and viruses. Biofilms are 1,000 times less sensitive to the same antimicrobials. We learned a little bit about biofilms already, but remember that they're an aggregation or association of many different types of organisms that are coming together, lots of different species. And they form this gel-like protective layer on their surface. And they start to communicate with each other to form these very complicated networks of microorganisms. And they almost function like a multicellular organism. If you use one antibiotic, there's no way that that one antibiotic can kill every microorganism in that entire bundle because it's an association of multiple species, multiple different types of microorganisms. Different phenotypes are expressed by biofilm bacteria. Biofilms, to treat, you have to interrupt their quorum sensing pathways. Quorum sensing is how they sense that other microorganisms are around and how they communicate. So if you cut off their communication and isolate them, then they can't function as a multicellular organism. Adding DNase to antibiotics. DNase destroys DNA, so that's a good way to fight that. Pre-treatment, so scrape off the biofilm and treat it, and then treat it a second time with a different antibiotic. Antifungals. So with antifungals, they're difficult to treat. The antifungals are difficult to come by because fungi are difficult to treat. Fungi are eukaryotic microorganisms. Eukaryotic means that they have all of the same organelles that are found in our cell. So when I talked about selective toxicity, this is where it's a problem. It's hard to find a target that will only kill the microorganism and not harm us. So some major categories include the imidazoles. So azole, when you see that at the end, you know that that's talking about antifungal. And the triazoles, one of the big ones is fluconazole. Fluconazole is given for the common yeast infections, like vaginal yeast infections. And you can see all of the different types that are listed here. But if you see azole and I'm asking for which one is one of the common antifungals, look for that term azole. Antiprotozoan agents. So there's a lot of different protozoan infections. The trypanosoma, leishmaniasis, the toxoplasmas, the giardias, the amoebas, and malaria. I'm going to focus on malaria for this discussion. So with malaria, quinine was one of the original drugs that was used. It was actually a fever-reducing tree. So it was this tree that if they consumed the leaves, they would get rid of their fevers. Well, today we know that the fever was caused by the malaria parasite, but back then they just thought that it was a standard fever. So they would consume the leaves of this quiona tree in order to get rid of their fever. Well, they heard about this in Europe, so some empress over there or some queen or something had called the people from South America and said, go bring me one of these quinoned trees. So they brought the tree over and they planted them all over Europe and they used it as an antimalarial treatment way back when. Well, that started to not work as well. And they came out with other types of drugs like chloroquine. Chloroquine is still used today. And it was recommended by the WHO and as one of the major treatments for malaria, but now they've switched to artemisinin. Artemisinin is a component of a combined therapy. So today, instead of just taking one drug to help treat malaria, you have to take two drugs at the same time. This is the new recommendation by the WHO. The therapy is called ACT. So artemisinin combined therapy. It consists of artemisinin, which is a very short-living drug, combined with one of these other drugs. So chloroquine, mefloquine, primaquine, pyrimethamine. Very rarely would it be quinine, and there's other drugs that exist. that you would put in combination with this. So the artemisinin is very powerful and very short-lived, and this is comes from a plant as well. It's just not a tree, it's more of a shrub plant. This is very short-lived. And then the second drug lasts for like 7 days. It has a longer life of activity inside of the body. So you give these drugs in combination, hoping that shooting it with 2 drugs will wipe out any of the malaria parasites. If a person gets malaria, it's likely that they're infected with 2 different species of malaria at the same time, or 2 different strains of malaria at the same time. So you could have one that's resistant to artemisinin and one that's resistant to pyrimethamine. So if you give them a combination of artemisinin and chloroquine, then that should wipe out everybody. Artemisinin is also being considered as an anti-cancer drug. So there's some associations between this drug and treatment of cancer and modulating of the immune system that makes it a prime candidate for cancer therapy. So they're starting to look into that for other types of treatments. So the drugs you should know for the antiprotozoans would be the ACT, which is artemisinin combined therapy. And you should know quinine. So quinine was the original antimalarial drug. Antihelmet agents. So if you have worms, or someone you love has worms, so a pet or something like that, you need to diagnose what the worm is before you choose an appropriate treatment. People that have dogs that want to deworm their animals and they go to Walmart or whatever and buy your standard dewormer, usually that's not effective, because the standard dewormer that's sold over the counter is usually just going to target the roundworms. So pyrantel is one of the most common drugs that's sold over the counter. If you diagnose the worm, you can know which drug to use to treat it. And with these antihelminths, a lot of times they're going to affect the reproductive capabilities of the microorganism. So they'll prevent replication, and eventually the worm that's inside will die of old age and go away. So mebendazole is kind of a broad spectrum antihelminth, antiprotozoan drug. So mebendazole works against a lot of different microorganisms. Pyrantel is specific for roundworms, and praziquantel is specific for flatworms. Praziquantel also has some effectivity against tapeworms. So again, diagnosing that infection will give you the appropriate drug to use. So those are the 3 that you need to know: mebendazole, pyrantel, and praziquantel. We learned about how to diagnose helminth infections way back in the beginning of the course, so hopefully you remember a little bit about that. Diagnose first and then treat with the appropriate antihelminth. Antivirals. So there's 3 major modes of action. You can bar penetration of the virus into the cell, you can block the transcription and translation of viral molecules, and you can prevent the maturation of viral particles. Why aren't there more antivirals? Because of the high replication rate and mutation rate of the virus. So we know that we have to get the flu vaccine every year, because every year there's a new strain that's most popular, or most likely to come in contact with people and infect people. It mutates. And as soon as it gets inside of the host cell, it mutates again. This is why HIV is so difficult to target, because it's constantly mutating. And when HIV gets inside of your cells, it's really just a strand of genetic material that's now incorporated into the DNA of your T cells. So it's hard to even find it, and then when you find it, it's hard to target it. So these drugs are few and far between, but a couple exist. Amantadine and its relatives, so zanavivir, which is Relenza, oseltamivir, which is Tamiflu, and then there's a new one, but these are going to be effective against the influenza virus, or influenza A virus. So no amantadine against the flu. Influenza A virus is now almost entirely resistant to this. So for example, Relenza doesn't work at all anymore, and Tamiflu is down to like 50% effectivity. We limit exposure of the virus to these drugs to prevent the development of resistance. We usually only give the drug within the first 48 hours of symptoms. Why? Because after 48 hours, the patient's just going to go through the entire infectious cycle anyways, so there's no stopping it at that point. We also limit it to critically ill patients. So if a patient is in high risk categories, so high risk of death, if they're immunocompromised or if they're elderly or really young and they end up getting the flu, then we give them the drug. But other than high risk groups and within the first 48-hour symptoms for everyone else, we usually limit the exposure of the drug to the pathogen. Acyclovir, so Zovirax or trying to think of the name of the herpes medication. Valtrex, there we go. So acyclovir, also known as Valtrex, so this is against the herpes virus. What this one does is it prevents replication of the herpes virus. This doesn't get rid of the virus, it doesn't cure anyone of this virus, but what it does is slows down the replication. So when you get herpes, the herpes virus goes inside of your neurons and it hides in the neural ganglia. And whenever you have an outbreak, it will start to replicate again. And when it replicates, it rides the nerve all the way to the end and then emerges toward the surface of the skin. And then you have that outpouring of blisters, usually in the same spot every time. Kind of like when you get shingles, and we'll talk about shingles later on, but shingles also hides inside of the nervous system, and then it rides the nerve up to the surface of the skin, and you have an outbreak of the blisters on the surface of the skin. So very similar type of action going on there. If you prevent the replication, you're going to decrease the likelihood that the person's going to transmit the virus and decrease the number of outbreaks. You can get herpes and never have an outbreak ever, not even a first outbreak or an initial outbreak. Or you can get herpes and have outbreaks every other day. So the people that get frequent outbreaks are going to benefit the most from this drug. Taking this drug does not mean that you will never spread this to a partner. And not having an outbreak doesn't mean that you're not going to spread it to a partner. So you can constantly shed it and spread it, and there's, even without symptoms, you can be passing this from person to person. But this definitely decreases the replication, which decreases the likelihood of transmitting it.